With DNA, there's only one type of DNA. And everybody's got the same type of DNA. Every organism has the same type of DNA. It's a sugar and a phosphate and a base. There's 4 different types of bases, but everybody's DNA comes in that structure, and it's a double helix. With RNA, there's not just one type, but there's 3. Well, this becomes confusing for some people, and they have a hard time remembering the difference between these 3 types of RNA. So this is another one of those instances where there's a process that takes place. We're going to look at proteins being synthesized, and in order to understand how that takes place, it's beneficial for people to draw this out, draw this process taking place, not just have a flashcard RNA, this type, and on the back you've got the answer, but to be able to follow this process of RNA being made based on the information in DNA, and that RNA being used to make a protein. In order to do that, you've got to use 3 different types of RNA. So that's what we're going to look at initially, are these 3 different types of RNA and what they do. They're all RNA. They've all got a sugar and a phosphate. The sugar is a ribose sugar instead of a deoxyribose sugar. And the phosphates are the same. The bases, there's 4 different types of bases. In RNA, there's a U instead of a T. So the first of these is messenger RNA, and then there's transfer RNA, and there's ribosomal RNA. 3 different types. Now, people sometimes don't go to that much trouble to make sense to themselves what the difference is between these, and there's only 3 of them. So if you spend some time looking at what each one of these is used for, then it's going to make a lot more sense than if you just say, okay, there's mRNA and tRNA and rRNA. They're all 3 different, but I have a hard time remembering what they do. Well, each one of them has a different structure. Each one of them has a different lifespan. Each one of them has a different role, but they're all 3 involved in taking information from DNA, taking that information out to the ribosome, and there making a protein based on information in DNA. That information in DNA is in a gene. So that's what we're going to see. We're going to look at the structure and the function of each type of RNA. There's only 3, so we're going to try and keep this simple. But the first one of these is messenger RNA. Messenger, what does that mean? It's a messenger. It's sending a message, taking the message from DNA out to the ribosomes. This is how you get from information in DNA, which doesn't want to leave the nucleus, doesn't want to break apart or do it. It doesn't want to be modified. It wants to stay in this protected environment and continue to store this information for later use. This messenger RNA carries the information from DNA out to the ribosomes. This messenger RNA is synthesized based on information in DNA, in fact, based on information within a gene. And then once it's made, there's some alterations that take place before it leaves the nucleus. But when those alterations are done, you can see these things being spliced out and the messenger RNA being modified. If you look at that figure, you make messenger RNA. It's got these blue parts and these gray parts in this diagram. These gray parts are spliced out based on what the cell is doing, the control of making that messenger RNA. Then you've got a final version of messenger RNA, that blue ribbon, that leaves the nucleus of the cell, goes out to the ribosomes, and it's got the information used to make a protein. It's got the information that it's taken from DNA. It's got the message of how to make a protein, and it's taken it out of the nucleus and delivered it to the ribosome. That's what messenger RNA does. It delivers the message. So this messenger RNA we're going to see is made, it's used, and then it's recycled. So some of this messenger RNA only lasts for a couple of minutes. It's made, it goes out to the ribosomes, it's used to make a protein, and then it's broken apart again and reused. That's messenger RNA, mRNA. There's also transfer RNA, tRNA. This is RNA that transfers the information from messenger RNA to a protein. This is the RNA. It transfers. Transfer is the name of it. Transfers information that's in RNA to message in a protein, to information in a protein. So you know what RNA is? It's a sequence of nucleotides. Sugar phosphate base, sugar phosphate base, sugar phosphate base. That's the information in RNA. What transfer RNA does is takes that information, nucleotide, 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 and transfers that information to a protein. Proteins are not chains of nucleotides. Proteins are chains of amino acids. So transfer RNA, we're going to see, transfers the information from a chain of nucleotides, that's what we find in DNA, that's what we find in RNA, to a chain of amino acids, which is what we find in proteins. That's what transfer RNA does. If you look at this transfer RNA molecule, it's got a whole bunch of nucleotides that are joined together. These form bonds with one another. It forms this three-dimensional figure. It's not just a chain of ribonucleotides. It's a chain of ribonucleotides that are bound together, and it has got a couple of important binding sites. Since this is transferring information from nucleic acids like RNA to proteins, which is a chain of amino acids, it's got binding sites for nucleotides. That's the anticodon region. Down at the bottom, you see those three pink red squares. That's a part of the transfer RNA that recognizes other nucleotides. That's where the bases form their pairs. Remember, ribonucleotides all have bases. That's where those bases are going to pair up. Another part of this transfer RNA molecule has binding sites for amino acids, where the bases are going to be used to recognize some amino acid. It's a very specific sequence of bases. There's this base, and there's that base, and another base. It's an A and a U and a G, or a G and a G and a C. That is going to recognize a particular amino acid. So there's a transfer RNA that's got an anticodon region of three specific bases. And the other thing that this transfer RNA has on it is an amino acid. It's formed a bond with an amino acid. So it's carrying an amino acid to the ribosome. Not just any amino acid, but one specific amino acid that's supposed to go in a certain location when this protein, which is a chain of amino acids, is being synthesized. So for instance, it's like making a train of amino acids where there's a long train, and each car of this train is an amino acid. Well, the first one has got to be the engine, of course. So there's one transfer RNA that's carrying an amino acid that's analogous to the engine. This one always goes first. And that's what starts the chain of amino acids that's making up a protein. And then the second one is the car that's carrying all the coal or water, whatever kind of train it is. That's the second one that goes. It's a very specific order. It's not an engine and a coal thing and then these passenger cabin cars, but it's a certain amino acid and then another amino acid and another amino acid. So when we look at protein synthesis in more detail, that's what we're going to see, is that these transfer RNAs are very important, and it makes this, it allows this transition between a chain of nucleotides and a chain of amino acids. They're two very different things in two different macromolecules. Okay, the third type of RNA is a ribosomal RNA, or rRNA. Now this has more of a structural function than being involved in these processes. It's a more long-term type of RNA, and they're found in association with the ribosomes. That's why they're called ribosomal RNA. So most of the RNA that you find in a cell is ribosomal RNA. It's different from messenger RNA, and it's different from transfer RNA in that these long-term RNA that's in association with these ribosomes. It's a structural type of RNA, and when we look at the ribosomes specifically, we're going to see that they've got this structure with a small subunit and a big subunit. Part of that is proteins and enzymes in that ribosome, and part of that is ribosomal RNA in those ribosomes. So this is going to be, it's not going to be as difficult to understand what ribosomal RNA does. It's a structural part. It's got these different parts of part RNA and part protein that make up ribosomes. Okay, well, let's start looking at this process of protein synthesis. You've got RNA, which has got information that has been taken from DNA. DNA is not doing anything. DNA is not an enzyme. DNA is not proteins. DNA is not a chain of amino acids. RNA, it's the same thing. It's not a protein. It's not an enzyme. It's not a chain of amino acids. We need to get from information in a nucleotide sequence, which is both DNA and RNA, to information, not just information, but an actual structure that's made up of amino acids. So that's what we're going to look at next, is taking information in an RNA that was synthesized based on information in DNA, and taking these other two types of RNA, ribosomal RNA and transfer RNA, and making a protein. Now, I know for a lot of people this is a lot of all these words that don't make that much sense. Ribosomal RNA, ribosomes, and all these nucleotides, ribonucleotides. But again, you have to think about this. You have to. It makes sense for people to map out this process one way or another, however you're doing that, drawing something, to demonstrate to yourself that you understand how you get from information in DNA to information in to a functional protein. Now, this is something that I've alluded to before. I've mentioned before that if you look in DNA, and DNA is composed of these nucleotides, deoxyribonucleotides. DNA is composed of a whole bunch of nucleotides, a sugar and a phosphate and a base. If you counted all the sugars and phosphates and bases, and you know they come in pairs and they're in this double helix, there's two chains that are complementary to one another. If you counted all the nucleotides, or in other words, all the base pairs in DNA in humans, you'd find almost 3 billion base pairs. So there's almost 3 billion times that you've got a G and a C and an A and a T. That's a lot of information. A lot of that DNA didn't do anything, but some of that DNA encodes information, and some of that DNA is used to make RNA, and a lot of it's used to make proteins. So there's genes that are found in that DNA. There's genes that make up part of those base pairs. And if you look in humans, there's as many as 25,000 different genes. So there, in other words, in DNA, there's instructions for us humans to make as many as 25,000 different proteins. Now that's a lot of different proteins that can carry out a lot of different functions. If you look at these other organisms, then we've got a lot of base pairs, 2.9 billion. We've got a lot of genes, 25,000. But if you look in some of these other animals, even some that aren't really that complex, like yeast and bacteria, they've still got millions of base pairs. They've got millions of sugars and phosphates and bases that are joined together in these pairs. It's a very large amount of information to make a very large number of proteins that can carry out a very large number of functions. Okay, so now we're going to turn our attention to this idea of taking information in DNA and making RNA. And taking that RNA, traveling out to the ribosomes with that RNA, and using that information in RNA to make a protein. Those are two different phases of protein synthesis. Transcription is where DNA is used to make RNA. Transcription is where DNA is used to make RNA. So now you've got RNA. It's not an enzyme. It's not doing these different things that proteins do. Translation is where the information in RNA is used to make a protein. So let's start with this transcription first of all. Let's start with making RNA based on information that's in DNA. And that'll be good for this lecture. So what do you need for RNA to be synthesized? Well, you need these different ribonucleotides, not deoxyribonucleotides, but ribonucleotides. You need the building blocks of RNA. You need a sugar and a phosphate and a base. And you need 4 different types of bases. You need some where the base is an A, and some where the base is a U, and some where the base is a C, and some where the base is a G. So there's different enzymes that are manufacturing these ribonucleotides in cells. You need to know how to put these ribonucleotides together. What order are these bases going to go in? What order are those ribonucleotides going to go in? Is it first going to be an A and then a U, or is it going to be an A and another A, or is it going to be an A and a G? What order are they going to go in? That information, that blueprint or set of instructions is in DNA. And it's based on these bases forming complementary pairs. You also need some energy to carry out these reactions, basically joining these ribonucleotides together. We looked at phosphate and in ATP and energetics and saw that there was a big role of phosphate. And you notice that every one of these nucleotides has phosphate. And you also need some kind of equipment to join these ribonucleotides together in the sequence that they're going to be joined. So you need some enzymes. And in cells, there's this enzyme called RNA polymerase. Polymerase refers to making something. What it's making is RNA. Okay, so that's what we're going to see when we look at this.